Kirti, can you hear me? Hi, Kirti, can you hear me?
All set up, huh? அக்கா லாகின் பண்ணிட்டீங்களா? லெஃப்ட் ஆயிட்டாங்களாப்பா? அக்கா எஸ். Shall we start, Akka?
Yes, Kirti. I seem to be having problems sharing. I can't share the screen. Kirti, can you hear me? Kirti, can you hear me?
Akka, yes, Akka. I'm able to hear you now, Akka.
Kirti, when I go on to present and your entire screen, the share button, that's not highlighted. So I don't have an option to share.
Akka, yes, you'll be able to share now. Just give a try now, Akka.
The blue share button, it's not highlighted. When I click on it, nothing happens.
Okay, so now what are the other options that you have, Akka?
I've got just cancel and share is not actually highlighted. So when I click onto it, nothing happens. Let me try. It says can't share your screen. Sorry, an error has occurred.
Shall we leave the meeting and rejoin again? Will that be all right? Shall we try that one?
I'll try that. I'll try that.
Not an issue, Akka. We will try to sort it out. But I'll send you a mail. Download பண்ணிடுங்க. Yes, Akka.
Okay, going on to your entire screen and clicking on and sharing, and it says can't share your screen.
Akka, not a problem. I have your PPT here, so I'll play that. Will that be all right, Akka? We'll do that. We have a backup of. Please don't worry. We will be able to sort it out. Yes.
Oh, brilliant! Thank you so much, Kirti.
Yes, Akka. Just give me 1 minute. We'll just open everything. Yes, Akka. So yes, it is getting downloaded and we'll be able to present from here and you'll be able to see. So that will
All
not be
right.
an issue. And if you want to go to the next, yeah.
When I want to move on to the next screen, so I've got
Okay.
to let you know, isn't it? Next screen, please, Kirti.
Yes, Akka. Just tell me, then we will move on to the next screen. Just say next screen and we'll move on.
Thank you. Thank you so much. I knew this
Yes,
technique
Akka.
will happen. We'll make do
Okay.
with it.
Yes, Akka. Yeah, we will start now the technical session. It's 5 and 6. Yes. And good morning, dear ladies and gentlemen. We are now moving on to the technical session 6. And so you'll be excited for another technical session, which will be chaired by me. I'm Dr. Keertana. And for our speaker, we are having Dr. Ruby Vijaykumar. She's from UK. And to give a brief introduction about her, Dr. Ruby Vijaykumar is a clinician with over 40 years of service. She did her Bachelor of Medicine in Stanley Medical College, Chennai. She completed her post-graduation in dermatology in Madras Medical College, Chennai. She did her MSc in Information Technology in University of Teesside. She is a member of Royal College of General Practitioners, London. And Dr. Ruby Vijaykumar completed her diploma in Practical Dermatology in Cardiff University, UK. She also completed her post-graduation in medical education. And she's a fellow of Royal College of General Practitioners of London. And with that, we are having a clinician, and we already had a clinician before in a previous talk, and she's another clinician who will be sharing her views and ideas of patients she's seeing with genetic disorders. Yes, we welcome you, ma'am. And just a minute, we'll be having you the presentation ready. And if you want to have an interaction with the students, you can give 1 or 2 words to them about how what is happening in UK and about the NHS services you wanted to give. Talk about
Excellent.
that.
Yeah, thank you very much for your kind words, Kirtana. Hello, good morning, everyone. I think it must be around 11 o'clock in India. It's 5 o'clock here in the UK, and it's quite bizarre sitting in my study, just looking outside, and it's quite dark. For the literature lovers, if you know a novel called Wuthering Heights by Emily Bronte, it was very much set up in West Yorkshire. So, so welcome to Yorkshire. I wish it was more of a daylight so I could just show you the Yorkshire, lovely Yorkshire moors. So there is a bit of a conflict here. I must confess, Keertana very nicely introduced me. I must say that she is my very dear cousin, and we know her as Kirti. So if I'm come across very familiar with Keertana, it's just because she's my cousin. So it must be about a year ago, and it did feel a very long time, somewhere in the real distant future, when she called me saying that, do you mind giving a talk? I thought, all right, yes, it's just 12 months, 15 months. And I said yeah, that's absolutely fine. We didn't decide on the topic or anything. And before I knew it, I had a very kind invite from Dr. Nidhi Rajan. And thank you very much for your invite. And thank you for asking me to be part of this international conference, Dr. Nidhi Rajan. I hope to meet you sometime when I come to India. So that's about an introduction about how I am here today. And if I could just, well, Kirti very nicely, she introduced me. The only thing that I have always been proud of is something that was not mentioned, which is I am a small town in Tamil Nadu, uh, and really proud of it. Yesterday, again, Dr. Nidhi Rajan kindly had sent the link to yesterday's event, and it just made me realize what a small world. There was one of the chief guests who is currently in South Korea, if I'm right. Again, from my small town, and it kind of made me happy to hear that he is from Salem. So there you are. I was born and brought up in Salem, and then did schooling. I moved to Stanley Medical College and then Madras Medical College, 1996. Joined my husband here in the UK, and I've been living here for the last 3 decades or so. So, I'd like to. Next slide, please, Kirti. Sorry, you'll be hearing a lot of this. Next slide, please. I'd like to talk to you a little bit about the organization that I work in, again, which I'm really proud of. It's the National Health Service. So, National Health Service in the England, it's very, very unique. Unique in the sense it is free for all at the point of use, and every resident in the UK, they don't have to pay. It's free of charge at the point of use. So that's what makes NHS so unique, and everybody who's part of NHS feels so proud about the whole organization. So
Akka, we able to see, sorry to interrupt you. We are able to see the presentation.
So what is NHS? So next slide, please, Kirti. So NHS is it starts off with two prongs. You have primary care trusts and the acute trusts, and then we come together under this strategic health authorities where all the policies and the standards and everything is set. Then obviously that's under Department of Health, and Department of Health, like every other country, is under Parliament. So the politicians decide what is good for the patients, not people like you and me. So that's pretty much like every country. But this is the general structure of National Health Service. Next slide, please, Kirti. So this, when Kirti asked me, about the topic, I would like to share an experience that I had a good while ago, maybe 10, 12 years. Um, it was quite emotional, and it stayed and it will stay with me forever. Like Kirti said, I'm a clinician, so it was like everybody. One fine Monday morning, I was there, sat in my clinic, when I saw an say about early 40s lady, and I know her well. I know her family. I know her son and her husband. So, as luck would have it, this lady came to see me completely broken. So, normally, a general practitioner's consulting time is about 10 minutes, but this consultation went a bit longer. And out of that 10 minutes, first, she just cried her eyes out for the first 5 to 7 minutes. There was no way I had to give her the time. There was no way I could console her and just start the consultation. The reason behind that was two people. She had the only son, and then obviously her husband. Her husband, I did a 2 week referral a few months back, and I had sent him for tests, and he was diagnosed with colonic cancer. Then a 30, just a 31-year-old only son came to see me with a skin lesion. And I just, really, I still remember how I felt that moment when I looked at that rash. It was quite obvious, even clinically, that it was malignant melanoma. So here we have a lady with two people, probably her whole world, both with cancer. Sadly, very sadly, over a period of time, I'm talking about within, say, 12 to 15 months, she lost both of them. So that's the sad story, and that is real life that we come across on a daily basis. So when Kirti came to me asking about the topic, and we were discussing, and I thought, right, I asked about the audience, and she said, it is my students, my colleagues. And I thought this topic will be really fitting, because I really wanted you guys to know that the global community, the patient population, they actually need you. They need your work. So I thought I should bring the story to you guys on behalf of the patients that we clinicians see on a daily basis. So that's why I wanted to choose this. I chose this topic, and I wanted to talk to you guys about it. Sorry about this. I've got a frog in my throat. So, genetics and cancer. So, WHO report says that cancer killed nearly 10 million people in 2020. Remember, we were right in the thick of the pandemic, and the cancer care pathways were a little bit functioning out of norm. And we know that cancer is the leading cause of death globally. And in the West, the cancer risk to general population is 1 in 3 and quickly moving to 1 in 2. So people will, at some point in their life, 1 in 2 people will get cancer. And in the UK, cancer causes more than, sorry about this, 1 in 4 of all the deaths in the UK. We don't have an 2021-2022 data, but this is what I'm talking about, the statistics. It's all up until 2019. So there are around 167,000 cancer deaths in the UK every year. We are talking about really 460 every day. That's a big number. And we all know, United Kingdom, in comparison to India, such a big country, what's the size of UK?
Dear students,
so even
please
in
listen.
This country,
She's talking something
the size
related to your
the
subject.
size of the
Why your
country
work
into
is important
consideration,
in
if we
world.
look
All that
at
she's
that
speaking,
number,
kindly
that's
listen.
quite staggering. And every 4 minutes that someone in the UK dies from cancer. I don't know how you guys feel when you see these numbers. I was completely flabbergasted, because you don't go looking for the statistics on a daily basis when you're just so busy with other things. But when I just started to prepare for this presentation, that's when I actually knew that every 4 minutes someone in the UK is dying from cancer. And that's really sad. We are supposed to be really advanced, with all the work that you guys do to help with patient care. So it highlighted that there's more work to be done. Almost half of all cancer deaths are lung, bowel, breast, or prostate cancer. Again, this is just keeping in with UK in mind. Almost half of all cancer in males are from lung, prostate, or bowel cancer, and in females, it is lung, breast, or bowel cancer. So you can see it's the lung and bowel are the commonest cancer in both sexes. Next slide, please, Katie. Having said all this, UK mortality is ranked lower than 2/3 of Europe. And that's saying something, isn't it? Yeah. So let's talk about the common cancers that we normally see in the UK. We did say that the 4 common cancers are lung, breast, bowel, and prostate. Now, if we look at lung cancer, it is the 3rd most common cancer in the UK, and it accounts to about 13% of all new cancer cases. And we know that all the smoking cessation advice and everything has been there only in the last 2 decades. But in the 1960s, smoking, it was some sort of a fad, wasn't it? So, but we are actually seeing the consequences of that now. And we also had all this asbestosis and the mining and everything that just contributes to it. So there are around 48,500 new lung cancer cases in the UK every year. And lung cancer, and there are around, this again, for me, the mortality is what it's just really important to actually highlight the amount of work that needs to be done. There are around 34,800 lung cancer deaths in the UK every year. And looking at the breast cancer, it is the most common cancer in the UK. And there are about 55,900 new breast cancer cases every year. And if you look at the number of deaths, it's 11,500 every year. If you compare that with the lung cancer deaths, 34,800 to 11,500, you might wonder, how is it that it's the most common cancer, yet the mortality rate is not as high as lung cancer? Again, the credit goes back to the NHS. We have a screening program that every single women after a particular age that they go through, again, being NHS, it's free of charge. So it's the early detection. That's what helps. And we know that it's directly related to the survival. So that's about breast cancer. Now, bowel cancer, again, it is the 4th common cancer in the UK. The number per year, new cases, it's 42,900, not great, still high. And if you look at the mortality, it is 16,800. If you compare it with lung cancer, it's not as high as lung going on, which really helps with early detection. Finally, we have prostate cancer. This is again about 52,300 every year, and it is the most common cancer in men. We see that a lot. We know that men don't like to talk about things like this. They leave it too late, but the awareness is there. Men are starting to come talk to their GPs. There are quite a lot of programs out there to increase the awareness in men. So all that is working. So there are around 12,500 prostate cancer deaths in the UK every year. Thank you. Next slide, please, Katie. So let's come to the cancers that are linked to genetics, your forte. We've got a long list, but if you look at that, especially we spoke about those four common cancers, and it's quite interesting to see how a few of them are linked to genetics, and that's why I thought it'll be more fitting to talk to you guys about it. So breast and ovarian cancer, colonic cancer, Cowden syndrome, gastric cancer, Gorlin syndrome, men, neurofibromatosis, Peutz-Jeghers syndrome, pheochromocytoma, retinoblastoma, von Hippel syndrome, and Wilms tumor. 10% of the breast cancer cases are thought to be hereditary, and the most common cause of hereditary breast cancer is in an inherited mutation. You guys might know about this, the infamous BRCA1 and BRCA2 genes. On average, a woman with a BRCA1 or BRCA2 gene mutation has up to a 7 in 10 chance of getting breast cancer by the age of 80. So you can imagine that is 70%. 7 in 10 of them actually have a chance of getting breast cancer if they do have this gene. Moving on to ovarian cancer, again, a variant in the BRCA1 and BRCA2 genes account for most hereditary ovarian cancers. About 15% of women, we are talking about 15 in every 100, who develop ovarian cancer have a variant in either their BRCA1 or BRCA2. And there are variants in the other, like the RAD51C, 51D, and BRIPI genes can increase the risk of developing ovarian cancer. But these variants are rare. A little bit about the bowel cancer. This is something we see a lot. My daughter, she again is a general practitioner, and we were talking about this cancer and genetics the other day, and she mentioned that in her list they have quite a few patients with Lynch syndrome, and that is really worrying in the sense that when you look at the prevalence, but it is good that it's been actually diagnosed and patients are aware of the diagnosis and what can be done about it. Okay, so the Lynch syndrome itself, a person who inherits Lynch syndrome will have a high lifetime risk of colorectal cancer, which we do see a lot, as well as an increased risk of some other types of cancer. It can be caused by variants in 1 in 4 different genes, like the MLH1, H2, MSH2, H6, and PMS2. The code for essential DNA repair protein. In all 4 genes, only one affected copy is needed. As you all know very much, this is a dominant pattern in families. You also have the MAP, another inherited syndrome that increases a person's risk of developing colorectal cancer, and the MUTYH associated polyposis, which is more often autosomal recessive. Now, this next one, I have actually seen patients of babies with retinoblastoma, and it really gets to you. It does when you see a baby, because we do newborn screening tests at 8 weeks. So part of that screening is to have a look at the eyes, and I have diagnosed, not diagnosed, seen patients referred and later who went ahead to have a diagnosis of retinoblastoma. So retinoblastoma, it is a malignant retinal tumor that affects young children. Mutation in the RB1 gene causes retinoblastoma. almost all cases of retinoblastoma occur due to variants in the RB1. Next slide, please, Kirti. So we spoke about all the statistics and the numbers and how it actually clinicians actually see patients on a daily basis. I see about 30 to 35 patients a day, and I am guaranteed to see at least one patient, minimum one patient with cancer. So when we are talking about that high numbers, so what is this NHS doing about it? Okay. So we have the NHS has come up with quite a few cancer pathways. So what is a cancer pathway? It is the patient's journey from the initial suspicion of cancer, starting from when you see the GP with the suspicion of cancer, through the clinical investigation, and then the patient diagnosis and treatment. So what happens when the patient comes in and sees the doctor, and the doctor suspects that probably this is cancer? Then immediately the patient is put on something called a 2-week wait pathway. What it means is that right from the time the decision is made to refer a patient to investigate for cancer till the patient gets the test, that should be done within 14 days. And that's the 2-week wait. So the 2-week wait appointment system was introduced so that anyone with symptoms that might indicate cancer could be seen by somebody who's an expert as quickly as possible, which is 14 days. The target for all cancer treatment pathways is for at least 85% of the patients to start their first treatment for cancer within 2 months of an urgent GP referral. 62 days might feel a bit longer, but we must remember that we are talking about a big organization, free of cost, and all those things. So what happens? So you've got your patient, you've diagnosed the patient with cancer, what next? So there is a big multidisciplinary team that works around this patient and makes sure that the patient gets the proper right care. So people who are involved in the multidisciplinary team are the GPs, specialists, that includes oncologists. Then once the patient is back in the community, you have the community nurses who are called the district nurses. You have the Macmillan who are the cancer specialist nurses. You have the occupational therapist who goes to the patient's home to make sure that they have everything. For example, you have your colorectal cancer patients, then you need to make sure that they have the commode and things like that. Are they in a bungalow? Are they in a house? Can they walk upstairs? Is there a stair lift? Everything that's being done by the occupational therapist. Then you have your physiotherapist and the social worker who makes sure that they have a good care package do they have their finances sorted, who's going to pay for their meal, how are they going to, so they take care of pretty much everything. So this whole team works around the patient after the diagnosis of cancer. So then not every story has a happy ending. So when there are patients who go into palliative care, so the palliative care pathway kicks in. So what is palliative care? It's a specialized medical care for people living with serious illness, such as cancer, but not exclusively cancer. It can be for patients who are in their end of life due to heart failure and those sort of things. Then finally, you have, when it comes to the end, it's just end of life pathway, where all you're doing is just making sure you stop the no active intervention, but keeping the patient just comfortable by giving them the anticipatory medication. And all this is done by that multidisciplinary team. So that's the cancer pathway. So what are the recent developments? This is just an ongoing thing, and this is not something new. We've always had cancer in the community, but all we know is it's just getting the numbers are getting bigger. So genetics, as you and I know, usually refers to the study of the single gene disease. However, genomics, on the other hand, it refers to the study of the entirety of the person's DNA, which including the genes and the DNA in between, which is the genome. So what did NHS do about this? So NHS came up with this genomic medicine service, which was launched in 2018, not too much in the past. Those of you who are really interested in this, I would direct you to this website, and there's plenty of material that you can read. It's quite interesting. This is just going to be in my PDP for my next appraisal. I was quite fascinated. So what did they do? The transformational 100,000 genomic project sequenced about 100,000 genomes, adding significantly to knowledge of cancer and rare disease. So this is what is really interesting, the rare disease, which we will talk about in a minute, and providing more patients with diagnosis. So this was the first step towards establishing a genomic medicine service. NHS had not done this before. And increasing the use of genomic information within routine. We are talking about routine NHS patient care, when the patient just walks into a GP surgery and they can talk about, ask questions, and the clinician is expected to give them some information, or at least to refer them to the appropriate people. And that's known as mainstreaming. So what was the aim of this project? The main aims of this project were to make genomics part of routine healthcare. Watch this space, guys. You will be surprised. In the years to come, we are talking about another 20, not even 20, I'm thinking of 10 years, like people walking into a GP surgery, walking into a doctor's consulting room, asking for, I would like an ECG, I would like a blood test to get my blood sugar, blood cholesterol tested. People are going to walk in asking for this genetic testing. That is the future, and that is you. And the other aim of the project is enhanced genomic healthcare research. That's where you come. Plenty of opportunity for the ones who are in this field. If this is your specialty, a lot of opportunities out there. And also uncover answers for participants. There are plenty of patients with symptoms and simply with no diagnosis, and just clinicians are doing symptom control. So we need to provide answer for these patients, and these were the aims of this particular project. Next slide, please, Katie. So what did they do? They looked at the genes of about 4,660 people from 2,183 families. That's a lot of families. But what did they do is, with rare disease who were early participants in this genome project, and they were analyzed using, this is the future, the whole genome sequencing. And they've done some brilliant work on that. The results showed that using this whole genome sequencing, that led to a new diagnosis of 25% of patients. So what we are talking about is 1 in 4 patients were out there with symptoms, suffering without a diagnosis. What that means is, if there is no diagnosis, we can't treat them. So we were actually letting people down letting general public down because we didn't have the knowledge. So that is where research is so very important. And what it did to this 25% of patients is amazing. So many of these patients had gone through years and years of appointments. The number of things that they must have undergone, the number of drugs that they must have been given just to as a trial, the number of side effects, just because they didn't have a diagnosis. And what a good boon it must be for these people. Now, again, this WGS enabled them to not only get a diagnosis, pave the way for treatment, and save on vital health resources. Remember, it's all free of charge. That's why the emphasis is so much on evidence-based, cost-effective treatment in NHS. So, for example, there was a 10-year-old girl whose previous 7 years search for a diagnosis had multiple intensive care admissions, over 306 hospital visits at a cost of £356,571. Just imagine, leave all the money out of it and just looking at a 10-year-old, a normal 10-year-old, what do we expect? Go to school, come home, have a good play, go to bed. That's normality for a 10-year-old. But here you had a 10-year-old girl going through 7 years. So we are talking about pretty much from when she was born, having intensive care admissions, 307 hospital visits. That's saying something, isn't it? So genomic diagnosis enabled her to receive a curative bone marrow transplant at a cost of just £70,000. In addition, predictive testing for her siblings showed no further family members were at risk. And what a relief that must have been for the parents. So the pilot study showed that this whole WGS can effectively secure a diagnosis for patients, save the NHS vital resources, and pave way for other interventions. Next slide, please, Katie. You really need to. Because next will be a real-life story. I'd ask you to take a couple of minutes to actually listen to what these two families went through. Thank you.
So it was a genome sequencing test to determine what type of cancer it was and if the cancer was genetic. And we found out that it wasn't genetic, which was a massive, massive relief.
That test was the key to everything. That solved everything. Oh, without that testing, it would have been a completely different story.
Yeah,
I mean,
we would have gone for chemo.
So we did the biopsy, we sent off material for the normal testing, the normal standard of care, and then we also at the same time sent off stuff for whole genome sequencing, some tissue, which now in England is also standard of care. And at Addenbrooke's, we offer to every child who walks through the door.
It was an emergency diagnosis, really, that she had the cancer, even though we didn't know what type. But obviously our consultant knew after doing the tests, and we've been blue lighted to Addenbrooke's.
Well, you think it's a nightmare, really. You don't think it's true. You just think you can't believe it's happening, really, more than anything.
And it's just a big blur, really.
Yeah. So Aubrey presented to us right at the beginning when we were starting doing whole genome sequencing and offering it as standard of care. So we sent her tumour off. Now, under the microscope, it was very difficult for the pathologist to tell us exactly what her tumour type was, and there were a number of possibilities. And that made treatment at the very start of her journey challenging to work out what was best. But quickly, with the whole genome sequencing, we got the results back. And what that showed was that her tumour was a specific type of sarcoma called rhabdomyosarcoma. And by looking at all of the information in her tumour, we were really able to tailor her treatment going forward once we got that information.
In Oliver's tumor, we found a mutation in a gene that's called PDGFR beta. It doesn't really matter what it's called so much, but once you've got a mutation in that gene, you completely leave the territory of cancer, and then what you think about is a benign tumor called a myfibroma. What was so astonishing about the results was this. The type of mutation was a very peculiar type of mutation that we could not have picked up with any other assay.
To have that testing, we didn't even need to discuss it, did we, really? It was
it was offered to us, and we literally instantly just said, yes, go for it.
It meant that me and Paul sort of knew we hadn't passed anything down to Albury, and it also meant that we know our son Harry, who's just about to turn 7, is not at risk of getting this cancer also.
The whole genome sequencing information, along with the pattern of where her disease was on scans, told us that chemotherapy was the best option for her and avoided her going through.
Thank you. It's just a little bit more about what they've been doing. So I'm just quite mindful of the time. We are coming to nearly the last 5 minutes here. So I'm just quickly going to go into the future of genomics. So what can we expect and what are the opportunities out there? If you move on to the next slide and the one after that, please. So what have they done in the UK? Come up with this Genome UK25 implementation plan. So in October 2022, just 3-4 months back, NHS England published the first ever genomic strategy, which is accelerating genomic medicine in the NHS, which sets the strategic direction and priorities over the next 5 years. The aim of this is to ensure that genomics will be at the heart of the sustainable NHS in the future and the next generation of healthcare in the NHS. So it's all about the next generation. So there are about 44 objectives. I'm not going to bore you with all those, but a few of them being mainly to provide rare disease and cancer patients with an accurate diagnosis earlier and more quickly, whilst also supporting the use and development of a personalized treatment. This will also include a Cancer 2 program, which will be using this artificial intelligence to analyze the genomic data alongside the digital histopathology and radiologic images, so that you can give an accurate diagnosis and speed the diagnosis as well. And also extending the use of the treatment, the pharmacogenomics in the NHS will ensure more patients get the right treatment at the right time and the right dose, so there are no side effects, there are no adverse events, improving the outcomes for the patients and reducing the number of these. Using genomics to better understand the pathogens and how they spread. We've all been through the pandemic, so we know what a devastating effect it can have, as well as the role of the person's genome in the response to the infectious disease. This supports scientists in controlling outbreaks. Enable the NHS to move forward from a system that primarily detects and treats illness to the one that utilizes genomics to predict and prevent ill health. So, coming to the last slide, next slide, please. So we started off by saying how it is all gloom and doom, this whole cancer. But I'd like to end with a happy note, how the whole research, what it can do. If you could really play this clip, and hopefully we'll get the audio this time. Technology is brilliant, isn't it? Problem. It's not a problem at all. Thank you so much, Preeti.
Thank you so much for your time. And if you're having some questions, maybe I'll message you. And
and you will be able to answer us?
Absolutely. And guys, if you have any questions, please do let Kirti know, and I'll be more than happy to answer your queries. Thank you. Thank you very much. Bye. Bye, everyone.
Thank you, Akka. Thank you so much.
Bye, Kirti.
Students, can you please be quiet? Only if you're being quiet, I'll be able to hear them. சார் அவர் லைன்ல இருக்கார் பேசுங்க.
Alyssa was only 12 years old when she was diagnosed with a form of leukemia that's 96% curable. She soon found out she was in the other 4%.
Here now, I am connecting through the mobile. Okay, I'm not with my laptop. There is some issues with the laptop, so you need to share the slides in order to continue. Yeah. Yes. Exactly. Exactly. Yeah.
Audience. Audience.
Yep.
Please.
Okay, thank you. Okay, good afternoon to all. I am so happy to introduce one of our chief guests, Dr. Satish Kumar Rajasekaran sir. He is working as senior postdoc researcher in Rishon LeZion Institute in Israel. He completed his UG degree in biotechnology from SRM University in the year 2006. He completed his MSc degree from Madras University in the year 2008. In his PG degree, he got university third rank, and he completed his PG diploma in molecular diagnostics from Alagappa University in the year 2009. In his PG diploma degree, he got university first rank, and he completed his PhD in biotechnology from Bristol University, Tanjavur in the year 2016. He received 2 patents from Korea, and he published 34 research papers in the reputed journals, and he is also first author in 23 reputed journals, and he also called invited speaker in the 4 national conference, and he gave 4 oral presentation and 2 poster presentation in the national conference, and he received excellent postdoctoral fellowship of ARO from Israel in the year 2020, and he also got JRF fellowship from Government of India in the year 2015, and he has academic collaboration with Chonam National University and Wudik University and Yeongnam University from Korea. And thank you to all.
Okay. Yeah. Thank you for the introduction, and as it is already delayed, I think I will straight away proceed with slides. So I don't see the So shall I begin? Or okay, fine. That's good. So before I begin, I would like to say that the topic which I'm going to present right now is quite not very close to the conference overall theme, but I'm sure that I'll try to keep you intrigued, and most of the topics are informational, so it might benefit everyone who is listening. So the topic here is what you see on the screen is probiotic mystery with the volcanic community of lactobacillus cells. So indeed, the topic is ambiguous, and it's a bit of it will be confusing because I will try to clarify each and every points when we go through our slides. So before we go into the research slides or some of the figures or the publications which we have authored, I would run through a few terminologies to make the picture more clear. So the first slide here, what you have is probiotics. What are probiotics? For instance, probiotics are live bacteria, live bacteria with a non-pathogenic and can be beneficial to the to humans. So nowadays most people or most patients consume probiotics as supplements. So what you see is here is probiotic is a live bacteria, and is beneficial. And when these probiotics form biofilms, I'll explain what are biofilms in the next slides. They are called beneficial biofilms. Yes. So here you will know what exactly is biofilms. Biofilm is something which it's a sessile community formed by a bacteria that attaches to the biotic and abiotic surfaces. So in simple terms, if the biofilm are harmful if they are caused by bad bacteria, and they are good if they are caused by good bacteria. So for human health. So if you see the figure here, it's an example of a dental biofilms. Biofilms are, in short, they are sticky substances where bacteria, a mesh of bacteria which are shielded from antibiotics and the host immune system. So the bacteria inside the biofilms are quite resistant to any immune cells or antibiotics. That way, it's very hard to remove the biofilms. For instance, the dental plaques. If this is a biofilms formed on the tooth. So this is an example of biofilm formed by Streptococcus mutans. So in short, to be simple, if a good bacteria forms biofilms, it is beneficial, and that is a good bacteria means the probiotics, and it can be harmful. The same biofilms can be harmful if it is formed by the bad bacteria or pathogens. Next slide. So the concept here, which we are working on, is to use positive biofilms or the biofilms formed by the good bacteria to mitigate the biofilms formed by the pathogen bacteria. So there is a competitive exclusion principle going on here. What happens is that if there is a bad bacteria in our system or in our body, we try to take probiotic supplements. When we take probiotic supplements, what happens is that the probiotic bacteria or the good bacteria which is in the supplements go and compete with the pathogenic bacteria, which has already creating problems in our system. So when there is a competition between a probiotic and pathogen, eventually the probiotics win. So that's what is the concept. They use positive biofilms, so the biofilms formed by the good bacteria, to target the pathogen biofilms. Next slide. So you see, probiotics are multiple applications. There are diverse applications for the probiotics. For instance, although I'm focusing on how to mitigate pathogen biofilms, there are several other factors or several other bystander factors which are affected by the use of by taking probiotics. For instance, several human genetic disorders are connected to the are regulated by the probiotic supplements, and how for instance there are lots of genetic disorders which are regulated by the probiotics. Yeah, this is just a glimpse of other applications that a probiotics can work. Next slide. So now into the topic. We are working with the Lactobacillus probiotic family, and for instance, Lactobacillus is a very popular probiotics which is already in the market. You know, if you go to the pharmacy and ask for Lactobacillus supplements, they give you, because it's already proven to be healthy and beneficial to everyone, especially the old age people. So there are a lot of Lactobacillus species like AKC, Plantarum, Bulgaris, Acidophilus, Rhamnos. There are lots, and we work with Lactobacillus plantarum, what is seen in the red box. So we work with Lactobacillus plantarum, and here on I will be explaining our results with Lactobacillus plantarum. Next slide. Why this is important? Because I have already mentioned that there are lots of over-the-counter probiotic dietary supplements. So if you see the red box, you see the name of the probiotics, Lactobacillus plantarum. So this Lactobacillus plantarum is also readily available in the market. Yeah, please. This is just to show you. Yeah, next. So why Lactobacillus plantarum? So I would stop by this slide and I'll just brief you what is the term called nomadic. The term nomadic actually gets its name from nomads. Nomads are nomads. Nomads are people actually, they don't have a fixed living space. They keep traveling. They keep traveling from one place to another. So those people are known as nomads. So similarly, Lactobacillus plantarum has the trait that it is widespread throughout the ecology. You can find Lactobacillus in plants, in mice, in soil, everywhere in the ecosystem. So that's why it gets the name nomadic. So among all the Lactobacillus species, only 3 of the Lactobacillus species are classified under nomadic. So there are 3 different lifestyles. You have free living, nomadic, and host adapted. But the nomadic one is, it has a lot of adaptational advantages. So that is the reason why Lactobacillus is specifically considered a very good source of probiotic, because it is available throughout the ecosystem. Yeah, next slide. So yeah. So straight away now I'm showing you some of our very important findings. You see all the findings in a nutshell. So we used Lactobacillus plantarum that was isolated from the cows. And in the laboratory system, what we see is that we saw 3 different unique novel phenomena which was not revealed or identified before. For instance, I have marked the phenotype 1, the isolates from the Lactobacillus, isolates from the cow, where exactly they were able to form cone-shaped morphology, cone-shaped colonies on hard agars. You can see the cone shapes. And then during cold response, they formed a lot of aggregations and a lot of cold shock bundles. What we call is that biofilm bundles. And finally, the phenotype 3. I'll explain about each and every phenotype and I'll show you the results. So finally, when the last phenotype is that when you expose this to low pH stresses, what happens is that it entirely changed the geometric structuring. You see, usually the bacteria are single, but in this case of Lactobacillus plantarum, what we see is that V shapes. If you see properly, there are a lot of V shapes. So V shape conformations, which was first time discovered by our group, and we have actually authored the paper in NPJ, Nature Groups. Yeah. Next slide. Next slide. Yeah. So what we identified is that you see normally the colonies of bacteria on an agar plate is flat. You hardly get to see elevated structures or aerial projections. In this case, we have an aerial projection. You have a cone-shaped morphologies when you grew the bacteria in pH 5.5. And what we understood is that everything is regulated by pH, is controlled by pH. When you increase the pH to pH 7, that is neutral pH, you don't see the cone-shaped, although you see the cone-shaped, but there is a tip abolishment, and there you see there is a change in the pattern. There is no coloration, there is no brown coloration, and there is this justifies that at acidic pH they are adapted to external stresses. They have brown coloration, and they are not vulnerable to any other furnace for the stresses. So this was one of the phenotypes. Although I'm not showing all the results, I'm just showing the major images. Yeah, next slide. So what we do here is exactly we use that pH 5.5 grown cone-shaped colony cells, and we stress them to by placing them in minus 17 degrees Celsius. What happens here is that it's a cold stress. When you apply a cold stress to the cone-shaped colonies, what we saw a new phenomena, which we term as the sacrifice for survival phenomena. So what happens here is that a lot of during cold stress exposure, a lot of bacteria come together to form biofilm bundles. What you see on that, and what we termed is called consolidated bundles. You see red and green bundles. The green is actually the live cells, live bacteria, and the red are the dying or the dead bacteria. So cold stress actually kills the bacteria, but somehow a certain group of subsets survive, and how they survive is because the dead cells or the dying cells are shielding them within the bundle, and then that's how they recover eventually after the stress. So it's a sacrifice for survival phenomena, first time reported by our group. So yeah, that's it. So we further try to understand the mechanism of sacrifice for survival mechanism, how it is regulated. And when during this we try to understand what could be the genomic pathway that might regulate this sacrifice for survival system. So what we learned is that Lactobacillus plantarum, as I said earlier, it is nomadic, and they have a specific set of heat shock protein genes, which is not found in other lactobacillus. So that makes Lactobacillus plantarum unique. They have a set of 3 sets of heat shock proteins gene, HSP1, HSP2, HSP3. So these heat shock HSP genes are not found in other lactobacillus species. So we used the mutants of heat shock proteins HSP1, 2, and 3, and found that HSP1, that is linked to cryotolerance, when this gene is mutated, we don't get the cold shock sacrifice for survival phenomena. If you see the bundles in the wild type, WT is the wild type. If you see the wild type, you have the mixed bundles where live and dead cells are there, red and greens are shown. But in case of HSP mutants, what happens is that most of the cells die. That makes the gene very important for the sacrifice for survival rescue phenomena. So we identified the gene that is responsible and is the candidate for cold shock stress response via this. Thank you. Next slide. Next slide. So there were these are some additional These are some additional parameters. If you see that so you see what happens is that it's not just the during cold shock response. What happens that certain signals also activate, and these signals or signaling molecules are present in the cell-free supernatant, and then this cell-free supernatant when we tested the cell-free supernatant, that these signals can induce these cold shock bundles in other bacteria as well. For instance, we work with Bacillus subclassis model also, because that's one of the common model we use for generating mutants. So in this case, when we tried with Bacillus subclassis, what happens, the signals from Lactobacillus plantarum was used to enhance the biofilms of Bacillus subclassis. So there is a co-species interaction taking place. There's a crosstalk between Lactobacillus plantarum and Bacillus subclassis. That makes what happens is that so far we were using only one kind of probiotic, using only Lactobacillus plantarum, but now we recommend the use of a combination of Bacillus subclassis, because Bacillus subclassis also is a potential probiotic, and when in combination, it is easy to fight the bacteria, the bad biofilms, which I talked about earlier. Yeah, next. So right now we are trying to illustrate the pathway and trying to find what exactly are the signals. Next slide. Finally, the most intriguing phenotype is still it's we have the phenotypes. So was the formation of V-shaped conformation. If you see the difference in pH 5.5 and pH 3.5, you see the cells There are only single cells in case of pH 5.5. But in case of pH 3.5, what happens is a cell takes a new form. Cells takes a new form, because if you note the probiotics was first coined in 1919 by Erich Mechnikov, as a Russian scientist, and he was the first one to identify probiotics in yogurt. And this was done with there was a bacteria called Bifidobacterium, which also had a similar Y-shaped structures. So there is something with the structuring of the probiotic, which is really important for its activity later. So here we demonstrate for the first time a V-shaped conformation, and what we see is that V-shaped conformation structure is a set of 4 undivided cells. But why that's happening is that's what we tried to elucidate further, and we found that, next slide. We found that these V-shaped cells triggered positive biofilms. If you see the biofilms, on the lower panel, you will see the biofilms on polystyrene surfaces. What happens is that at pH 3.5, you have a thick biofilm stained with cyto9. That's why it's green, fluorescent green. In pH 5.5 and 7, you don't see a lot of green. That means there is less bacteria, so there is less biofilms. So what you see here is at pH 3.5, when the cells form the V-shaped structure, it actually triggers biofilms. So this is a new phenomena of generating positive biofilms via V-shaped conformation. Next slide. So next we try to find the significances of the V-shapes. What happens is that the V-shape, we try to do a competitive assay with a pathogen. We used Candida albicans. It's a human pathogen you have in dental biofilms and several other. It causes, it's a serious yeast infection. So we actually co-cultured. Try to study the interaction between Lactobacillus plantarum and the V-shaped structures and Candida albicans. So when we understood that the V-shaped structures were significantly eliminated the biofilms from the Candida albicans. You see, in the control of Candida albicans, you have a dense green biomass, but in case of Lactobacillus treated with 3.5, you don't see Candida albicans biofilm. So we have proved our concept that positive biofilms, a newly generated via the V-shaped geometric structuring, is able to remove the biofilms of Candida albicans and can be used as effective probiotics in near future. Next slide. So next was to we did this. We just scaled up the model and we used C. elegans. C. elegans is a nematode. It's a nematode which can be used for toxicity testing and other research parameters. This is economical because instead of going to the mice model, you can work with the C. elegans. So one interesting approach is that Candida albicans, when you treat Candida albicans or feed, exactly that's the term to be used here. If you feed Candida albicans to the C. elegans, the nematodes, the pathogen will kill the nematodes within time frame. But at the same time, if you co-culture with the V-shaped structure, that's what we did here. What happens is that it is able to prolong the lifespan of C. elegans. Lactobacillus, the V-shaped cells are able to prolong the life cells of Candida albicans infected nematodes. So this is a proof of concept that that our V-shaped structures are working fine in a living model. Now next we can always recommend for the human model. Yeah. Next slide. So straight away we are going to the conclusion. Actually, we in this talk, I have briefed about some of our various three different phenotypes which we identified, and one of them, the V-shaped structures, can be used for has a lot of applications. For instance, biofilm inspired encapsulation technology to bring up symbiotic food. Symbiotic is a combination of probiotic and prebiotic structuring. So there are a lot of applications of V-shaped structures. You can even lyophilize it and provide as a normal supplement. You can combine with Bacillus subtilis, use as a co-supplement with two different probiotics, and they're an option. So right now we are working on a similar pattern with Bacillus subtilis. You can see the cone-shaped structures that Bacillus subtilis erases or forms on agar plates. This is actually sort of a novel finding, and we are now trying to knock out the mechanism which is closely linked to the Luxus mechanism. Luxus is a quorum sensing mechanism. Okay, thank you very much. And with this, I should end the slide. And I thank all the organizers. I thank Dr. Nidhi Rajan, sir, and Kirtana. Thank you for your constant support. Thank you for giving me an opportunity. Thank you, everyone. Hope it was useful. Yeah. Thank you. Yeah, please shoot. Yes, you can exactly type it. Yes, please.
Satish?
Yes.
It was a nice presentation. Where are you now?
Sorry?
Where are you now?
Right now, I'm still in Israel. Actually, I have postponed my travel to US. I'm supposed to travel in 2 weeks' time, so hopefully I'll be coming by to India for a short visit.
Yeah, okay. Nice presentation. Thank you, Satish.
Yeah, please, instead of typing, maybe it's in the mobile, so I'm not able to see where the question is. Maybe if you can ask, I will reply. Yes? Yes, exactly. Though they are actually undivided cells. So there is an arrest in the cell cycle, which we are trying to figure out, which has some links to muramidase enzyme. There is an enzyme called muramidase, which is responsible for the final removal or final division. So there might be the V-shaped cells are compromised, or they do not express a lot of muramidase, and that's the reason why compromised, because it's a under low pH. When under low pH, it's possible that the enzymes are not expressed, because low pH is also a stressful condition. So they are connected but not divided. So it's still a single cell. Even the entire V shape, even if it is not divided, it's still a single cell. So the possibility is that the V shapes has 4 cells within. So when it divides, second generation will have 4 cells, but in the case of the single cell, what you have is once it doubles, you have 2 cells, but here you have 4 cells. That's very progressive technique to what do you call develop a lot of biomass. Thank you. Yes, exactly. The whole cell is one cell. Unless it is divided, it is not considered as a cell. Exactly. It's not. It's new to everyone. This finding was done last year, and yes, it's new to the scientific community actually. Thank you very much. Yeah, that's nice. Thank you very much. Thank you very much, and it was a great pleasure giving a talk. Hope it was useful. Yeah, sure. You can always mail me. Thank you very much. Bye. Have a good day too. Bye.
மா நான் தயவு செஞ்சு ஸ்டூடெண்ட்ஸ் தெளிவாக கேட்டுக்கோங்க. 1:45க்கு வந்ததுக்கப்புறம் வேல்டிக்டரி ஃபங்க்ஷன் நடக்க போகுது. அமைதியாக தயவு செஞ்சு உட்காருங்க. ஓகே, நீங்களும் டென்ஷனே எங்களையும் டென்ஷன் படுத்தாதீங்க. அமைதியாக 2 மணிலேருந்து 3:45 வில் ஃபினிஷ் த ப்ரோக்ராம். தட்ஸ் இட். ஓகே, ஸோ ப்ளீஸ் ஸ்டே சைலண்ட் டில் தட் டைம் அட்லீஸ்ட்.
Hello.